announce it on YouTube. It's okay.
I just started the stream and I'll start the webinar now.
Good morning, everybody. Hi, Julie. Looking forward to your talk. So, good morning, everybody. As we've been wont to do for the last few weeks, we'll wait another minute or two before we get started. Julie, do you feel like you need to try and share your screen just to see if it works? Or
I definitely need to try that. I've been having some unfortunate difficulties this morning. All right, what do you see?
Nothing yet. I mean, we see you.
Huh.
Yeah.
Okay, that's bad.
Looks like it's going to work.
Something's happening.
How's that?
Yep.
There you go.
Cool. That's great. That's great. Really fun stuff. All right, let's see. All right, we'll wait one more minute and then we'll get started. And thanks for joining us this morning. It's so nice to see so many friends from our many years at MBL and elsewhere. It's fun to see people tuning in, at least if I'm not mistaken. For example, Kinneret, so hi. I'm guessing you're in Israel. It's great to see you. Maria Jesus, I don't know if you're in Dresden or what, but hi to you. Good morning, Nathan, Avish. Wow. Hey, Ravi, Ricky Garner, Roya. Wow. Good morning, Sri, Steph Weber. Look at them all.
Hey,
Cool.
Julie, do you mind if we record your talk today?
Oh, that's fine.
Okay, thanks.
Do you mind if I introduce you?
I think you're going to, regardless of what I say.
Yeah, I know. It's a little scary. Okay, anyway, good morning. We're going to get started. So welcome to today's lecture, which ends the second week of Physical Biology of the Cell 2020 edition, which is very truncated, but it's been a great pleasure, at least for me. I'm going to start out with the facts of today's speaker. That's Julie Theriot from the Department of Biology at the University of Washington. So I'm guessing that most of you know Julie, but you know, one of the things that is a little bit of a secret, I would say, is that as an MIT undergrad, she wasn't just a biology major, but she was also a physics major. And what I would say, having spent the last 20 years, I don't know, I think we spend about 5 weeks or 4 weeks together every year. Her physics is really rock solid. After MIT, she went to UCSF, where she worked with Tim Mitchison and did really some super important work, which, you know, if you go to Google Scholar, you'll see that the papers that she wrote in the early 1990s are really highly cited, and they're also a pleasure to read. From there, she went to the Whitehead Institute for roughly 5 years, I think, where she was one of those heavy-duty independent postdocs. And from there to Stanford, where she spent, I don't know how many years, but quite a few. I'm guessing maybe it started to approach 20, I'm guessing, something like that. And then a couple of years back, moved to the University of Washington. And it's been really fun to see her after the move. And, you know, this is something that for the young people to think about, that there's really great virtue at times to changing gears, and it's very stimulating. So a few other little factoids. So probably many of you have noticed this, but Julie is the voice of the videos that you'll hear on the Alberts books. And she's, like yesterday's speaker, she won the MacArthur Award. She's an HHMI investigator. The one I'm most jealous of, frankly, is she got this special thing from the NIH. I don't know what it's called, but basically they think you're sufficiently excellent that they give you 10 years of funding, right? Do I remember that correctly, Julie? Something like that. So that thing's really great. Her work, she's made a dazzling array of discoveries in all sorts of different areas, and I'll just tell you a few that have meant a lot to me. So I came to know of Julie and her work through her work on listeria, and I have to say it was really the biochemical reconstitutions that I liked the most, specifically beads that, if I remember correctly, were covered with ACT-A, and then they would spontaneously lead to motions that mimicked the motion that you see with listeria. I thought that was really beautiful. The confluence of both biochemical studies and then also in vivo studies. You know, there's some famous movies that all of you have surely seen. And then there was a series of papers that she did with Dan Fletcher and Marlene Dogterom, where what they did is they looked at the polymerization, I would call it the polymerization motor, either microtubules or actin crashing into things, either using a cantilever, optical traps, and things like that to measure the force generation. Quite beautiful. And then there's a whole series of studies on cell shape that are also really, really inspiring. And, you know, for us in particular, the PBOC author team, you know, we, as I said, we meet for 5 weeks a year. We sit around, you know, day after day after day. And one of the things that we have a lot of fun talking about is coarse graining and what are the natural variables of a problem and how do you think about shape space and, you know, principal component analyses and stuff like that. And then I'm going to comment on two of her works with our speakers. So one is with Lena Koslover, and I don't know if Lena's here this morning, but she and Julie did a paper that I really love that has to do with cell division by fracture. And I recommend you go read that paper. And then the stuff that she did with Steph Weber, who I do see is here this morning. So just a few final words about the person and the intellect. So, you know, one thing that if you get to know Julie well, you'll see is that she has a formidable memory. And, you know, I really mean formidable. Another thing is that she can listen to others and instantaneously zero in on what they are doing, what the key questions are, you know, what are the outstanding issues, how to think about it from a modeling point of view, from an experimental point of view. And, you know, one of the things I would say about her is, to paraphrase, she really knows how to make a measurement, and we have a lot of fun talking about that too. One of my favorite pastimes that the PBOC author team knows very well is that much of our book is written with Julie pacing and me typing. And it's one of my favorite things to do is to have her pace. And I have to say that both Ernan and Christina have learned how to do this as well. And I enjoy that thoroughly. I think they've been taught by the master. They're still stumbling around, but getting better at it. Ernan and I did it yesterday, and I hope we'll do it again today. So finally, you know, she has a great joy of discovery. There's a playfulness actually in her science that that for me, you know, as somebody who's really intimate with her intellectually, you know, I see all the time, and that's a great privilege of interacting with her. So anyway, you know, one of my greatest friends, one of my greatest collaborators. I'm so happy to have Julie talk to us today, and looking forward to hearing what she has to say. So there you go.
All right, thank you. So thanks everybody for coming. I'm really looking forward to giving this talk this morning because I'm actually presenting work that I haven't presented before about shocking secrets of cell migration. Most of what I'm going to talk about is of a current graduate, Andrew Kennard, and a former graduate student, Greg Allen. But I'll also feature a few results from current postdoc, Nathan Beliveau, who is a graduate student with Rob. So I was inspired to do this, and actually I just changed my title, I think the day before yesterday, in part based on Carolina's talk last week about osmotic shock and the of osmotic shock and behavior of the microbiota in the gut. And in my own favorite problem of cell migration, osmotic shock has also been shown to play a really important role, particularly in triggering polarization of cells and also in altering their ability to protrude lamellipodia as they move. But there's another kind of shock, of course, that also we're all familiar with, which is electrical shock. And that's illustrated down here on the lower right. That is not a picture of either Andrew, Greg, or Nathan, but you could certainly imagine it applies to any of them. Because what they've been exploring and have explored is the ways in which electric fields in particular interact with the processes of cell migration and wound healing. So just to put this in a little bit of perspective, one of the reasons that members of my group and I have so long been fascinated with cell motility is because this is, I think, an absolutely fantastic example of a process where molecular mechanisms and physical mechanisms have to be intimately integrated. So in these very nice cartoons from a review by Laurent Blanchefant, you can see over on the left, the vision of a migrating motile animal cell as being made up of lots of different subcellular components, including actin filaments, myosin filaments, various different kinds of molecular machines that do things like connect the stress fibers to the substrate, protrude filopodia or lamellipodia at the front. But at the same time, you can think of this exact same cell as being a collection of physical elements that have to actually be able to generate mechanical force in order for cells to translocate across their substrates. And so equally, in terms of thinking at actin filaments and myosin filaments and all that, you can also think of the cell in terms of its elastic elements, its viscous elements, and the viscoelastic coupling between them that all have to then interact with physical environments with physical influences of the environment in order for a cell to actually go anywhere. So this problem of actin-based motility is one that's fascinated me for a long time, and it really is a great place to explore this connection between molecular mechanisms and how they all play out in order to get biological behaviors. Now, the particular form of cell migration that I'm going to talk about today, which is polyene motility that's based on assembly of actin filaments, this is something that is found really throughout all of the major branches of the eukaryotic tree of life, and seems to have a fundamentally conserved molecular mechanism that goes back to the early days of our origins as eukaryotes. Now, in animals in particular, cell migration gets a lot of attention because it's important for a lot of processes that are very close to our hearts, including things like development and the immune response, as well as wound healing, which is what I'm going to focus on really today. Cell migration is also very important in the context of cancer metastasis. So for example, carcinomas, which are cancers derived from epithelial tissue, typically are not life-threatening as long as they remain in situ, as long as they remain in the location where the cancer initially formed. But as soon as those tumorous cells acquire the ability to migrate and move away from that initial site of tumor formation, metastasize throughout the body, that's when most carcinomas become life-threatening. So at some level, understanding the mechanisms of cell migration may be useful for understanding all these larger processes, but it's also, I think, just a really beautiful and fascinating process in and of itself. And that sort of fundamental mechanism of cell migration is really what many members of my group have been focused on over the last, almost 30 years now. So understanding the mechanics and how the mechanics connect to the molecules certainly is intrinsically interesting. But what I'm going to talk about today is actually a little bit of a twist on that, which is not just how cells move, but how they actually decide where to go. So I'm going to show this movie now, which those of you who have ever heard me give a talk before have probably seen me show before. This is my very favorite movie ever. I think it's the greatest film of all time. It's like the Citizen Kane of cell motility. And this is a movie that was made by David Rogers at Vanderbilt University back in the 1950s, where what we're looking at is a blood smear, probably between two pieces of glass on a microscope. And this big creature in the middle is a white blood cell, a human neutrophil. And all of these are red blood cells. And then this little fellow over here is a bacterium. And based on its appearance, it's probably something like a staphylococcus. And when the movie plays, and I want you to realize that this is playing approximately real time. It might be speeded up by maybe 2 or 3-fold, but not more than that. What you see is that this neutrophil knows where that bacterium is, and it's arranging all of these mechanical elements within it in order to provide cell motility, so that it's able to chase after that bacterium with this single-minded ferocity, where its goal is to be able to phagocytose the bacterium and thereby help to prevent infection. So in the particular context of something like a neutrophil chasing a bacterium, it's thought that the major kind of information that the neutrophil is using to figure out where to go is chemotaxis, that is the detection of chemical signals in the environment. And in this particular case, it's because the bacterium is secreting formylated methionine peptides, which are the form of methionine used to initiate translation in bacteria that's not used in eukaryotes. And so when a neutrophil actually detects, using a G protein coupled receptor, when it detects the presence of peptides carrying formylated methionine, it knows that that peptide arose from a bacterial invader. So chemotaxis has been very well studied. We know a lot about the receptors and about all the signal transduction that connects the chemical signaling that's happening at the surface of the cell down into the underlying molecular machinery that's actually allowing the cell to move. But there's lots of other things that cells can respond to in terms of determining where to go, besides just this detection of soluble chemical signals. So for example, there's also haptotaxis, where instead of having G-protein coupled receptors that detect chemical signals that are floating around in solution, cells are also able to use other kinds of receptors to detect signals, chemical signals that are bound to substrates, for example, components of the extracellular matrix. Most cells are also capable of doing durotaxis, which is to evaluate the stiffness of their environment, and cells prefer to move towards stiffer regions rather than softer regions. They can also do plithotaxis, which is rather than responding to stiffness per se, they respond to sort of lines of stress that are present in their environment, either in the extracellular matrix or in their connection with other cells. Many cells, yes?
Julie, that makes me wonder whether durotaxis really should just be a subset, and really what we're saying is that you have to acknowledge that stress is a tensor.
Right.
In other words, do you think that's right? In other words, it just seems like the durotaxis case is a special case of you have a generic state of stress and then the cell cares. And for particular special simple forms of stress, maybe you would call that durotaxis. I don't know. I'm asking you. I'm not
yeah, no, I think in all likelihood that's probably right. I think for both durotaxis and plithotaxis, the work that's been done really so far has been to sort of identify what the mechanisms are the cells are using to sense changes in the environment. And they tend to have to do with adhesion. So it could well be an overlapping mechanism. I think in order though for durotaxis to really be a form of plithotaxis, it would have to be that the cell is able to sort of evaluate variation in substrate stiffness across across a single cell diameter and actually evaluate the gradient. And I don't know that mechanism has actually been shown, because there's other mechanisms you can imagine. For example, that cells, for example, they crawl around, and if their net rate of migration is different on a stiff substrate versus a soft substrate, you could end up enriching them on the stiff substrate without them necessarily being able to sense the stress tensor per se.
Yeah,
but
we had a couple of other questions if you're up for it.
Sure.
Yeah, so the first one is really fun, which is to note that the neutrophil and the bacterium are at very low Reynolds number. So how does the neutrophil ever catch the bacterium rather than just pushing them more? So that's really fun. Yeah. And then the other one, how is the mechanism for chemotaxis different from haptotaxis? So there's two more thoughts for you to address.
Okay, so the issue about low Reynolds numbers really is definitely right on. And in fact, if I could just go back and play this movie again, I want you to note that this bacterium is not flagellated. This is not a bacterium that can swim at all. So any movement of the bacterium that you see in this movie actually is coming about just because of that issue, with the fluid flow. So the neutrophil is actually pushing the fluid around it as it's moving. And you can see because of that, it's sort of bouncing the bacterium ahead of it. So it's really having sort of a hard time keeping up. And that's partly something that's imposed because of the geometry of this particular experiment, but it's also a real issue these cells have to deal with. And you could see when it finally caught up to it, it sort of, it almost sort of snuck around it and came at it from both the top and the bottom, I think in part to avoid that problem. And, really the question of how this works, what the timescales for this are in vivo, when you also have a lot of other sort of complicated issues in terms of movement of interstitial fluid, that I think is something we don't really have a good answer to. But the fact that this is super low Reynolds number also means that at every moment in time, you have to be constantly applying more force as a cell if you want to crawl anywhere. You don't have any inertia, so you can't just sort of, kick off and move. You have to actively be constantly applying force. And that's actually been very useful, I'd say, in the dissection of mechanisms here, because in some sense, the cell is always at a sort of quasi-steady state as it's undergoing motility. So those are definitely important considerations. It's also, I think, certainly true that most of us don't really have very good intuition about what low Reynolds number effects are really like. So I'm impressed with whoever asked that question, realizing that was actually going to cause the cell to push the bacterium away. That's definitely happening. And then what was the second question, Rob?
The distinction between chemotaxis and haptotaxis, I guess.
Oh, yeah. So, I mean, honestly, the reason that I'm making this list is because I love all these words, and I just wanted to sort of share the joy of all of these extremely specific words that are used to describe extremely specific things. Right. Really, the distinction between chemotaxis and haptotaxis comes down to two things. One is sort of the nature of the receptors. So for chemotaxis, these are typically G protein couple receptors. So they are able to, sort of bind to small molecules and then initiate signal transduction through heterotrimeric G proteins. For haptotaxis, the receptors, if you will, tend to be adhesion molecules. So they're things like integrins. So the nature of signal transduction is different. The reason for the different names, though, really comes down to the fact that it's just you do different kinds of experiments. So for chemotaxis, you put your chemical in a pipette, or you set up your chemical in a gradient, between two reservoirs. And for haptotaxis, the way you set up your experiment is to have the thing that you want the cell to detect in a physically bound form to the substrate. So these are really, sort of historical terms. I'm sure the cell doesn't actually have a strong opinion about, whether the substrate it's detecting is soluble or substrate bound. I think that's something that humans impose on the cells. But historically, those are names that have drawn that distinction between soluble and insoluble.
One more question about the movie. Maybe you mentioned this, but is the bacteria actually moving away from the neutrophil?
No, the bacteria,
No, okay.
It's a non-flagellated bacterium that has no intrinsic motility ability on its own. So all the movement you see away from the neutrophil is because of the fluid that's being pushed by the neutrophil as it's trying to crawl.
Got it. Thanks.
All right. Okay. So I think we got up to plithotaxis. Then there's also thigmotaxis, which is response to touch. There's phototaxis, which is response to light. Obviously, that's more important for photosynthetic organisms, but actually even most motile animal cells will respond to light if you shine it down a microscope, although that might be a temperature effect as well. And I left off thermotaxis. So there's also thermotaxis. So many cells will also respond to gradients of temperature. The one I'm going to focus on, though, today is my very favorite, which is called galvanotaxis, or electrotaxis. And this is directed movement in an electric field. All right. So it turns out that basically any cell that's able to do actin-based motility is also able to respond to a DC electric field and crawl in a particular direction. Most cell types crawl towards the cathode, but there are some cell types that move towards the anode. And furthermore, in some experimental systems, including in dictyostelium and in fish keratocytes that we'll talk about in more detail, it's been shown that you can actually take a cell that intrinsically normally likes to go towards the cathode and switch it so that its preferred direction is towards the anode by putting on drugs that interfere with signaling pathways. So this is sort of an indication that it's not just that the cell is somehow physically responding to the presence of an electric field, but whatever the detection mechanism that's allowing the cell to know that there is an electric field present is coupling into the same sort of signal transduction pathways that are also used in all of these other directional sensing mechanisms, at least one of them, the PI3 kinase pathway. So typically people give credit for the first description of galvanotaxis or electrotaxis to a paper in, sorry, that should be 1896, not 1986. Let me just fix that because that's actually much less impressive than the reality. 1896. There we go. And like I said, basically, any cell that will do any form of directed motility will respond. So this picture here is actually from a 1951 paper on Physarum polycephalum, which is this absolutely fabulous, enormous acellular slime mold. And this just shows a bunch of little explants of Physarum that were put on an agarose pad, and then an electric field was put across them. So the negative sign indicates the position of the cathode. And as you can see, the Physarum, which is crawling out over this agarose, is crawling in a very, very persistent directed way towards the cathode. The cells that we work on typically are much smaller than Physarum and individual cells. And for the most part, my group is focused on trying to understand migration of extremely rapidly moving cells. And that's basically because I'm a very impatient person, and so I like to see things go fast. So one of the fastest kinds of animal cells known are these epidermal keratocytes that come from the basal layer of fish skin, and they have a very strong galvanotactic response. So Greg Allen, when he was a graduate student in my lab some years ago now, started playing around with the galvanotactic response of fish keratocytes and set up a couple of little devices in the lab so that he could put cells on the microscope and then apply an electric field across the cells as they were on the microscope so we could watch this process in real time. So this shows one of his early movies. This is fairly low magnification, so each one of these little things is a cell. And I bet you'll be able to tell which way the electric field is going as soon as I start playing the movie. So as you can see, these cells are all responding really very, very strongly to the electric field and moving not perfectly parallel to the field lines, but but pretty darn well in that direction.
Julie, can I ask a couple of questions?
Sure.
Okay. So one of them is, what's the physiological relevance of the electric field sensitivity? And I think I actually asked you that last time I visited you. And the other one is related in a way. Does the cell migrate towards the cathode because of the charge of the plasma membrane lipids, because PI3K regulates PIP2 and PIP3 levels? So anyway,
okay, these are great questions. So the question of physiological significance is one that has inspired great debate. The conventional wisdom, the argument that has been made, is that one of the places where you're most likely to see a sort of large-scale DC electric field in living animals is after there's been a wound that cuts across an epithelium. And I'll go into more detail about this later, but the basic idea is that whenever you have an epithelial layer of cells that separates the fluid in two different compartments, almost always those epithelial cells have distinctions in terms of which ion channels and pumps they express on the apical surface versus the basolateral surface. And almost all epithelial cells put a huge amount of energy into carefully regulating the ion balance from the apical face to the basolateral face. And sometimes this is for processes like in your kidney, having to do with sorting out various toxins to filter out into the urine and things like that. But really it applies to pretty much any epithelium. There's always an ionic imbalance between the apical face of the epithelium and the basal face of the epithelium. Now, when you wound an epithelium, when you cut across this cell monolayer, which normally acts as a resistor, that means you basically short-circuited that separation of charge. And so you have a flow of charge, which is automatically going to give you an electric field. And depending on the orientation of that charge gradient, that could in principle be a directional signal to tell cells in the environment where the wound is located. And for most cases when we're thinking about a wound that will connect, that will short circuit sort of external environment from an internal environment in an animal, most of the time the effective cathode will be the location of the wound. But I'm going to show you more detail about that later as I try to convince you that this actually does have physiological relevance. But that's a very important question. There have been actually a lot of sort of medical devices that have been sold where you buy some little object that you put over your wound that imposes a magnetic field or an electric field. And there's some clinical evidence that some of those devices actually do seem to enhance wound healing in cases like diabetic ulcer formation in the skin. That's been very sort of controversial, and it's one of those things that some physicians swear by those devices and some think they're just snake oil. And I think part of the reason for the skepticism around that, even though some of the clinical data actually is pretty decent, really is because it's hard to imagine a molecular mechanism by which electric fields would actually promote wound healing. So I hope by the end of the day today, oh boy, okay, if I'm able to get through any of my data slides, I hope you'll at least have an open mind that might be a real possibility. As far as what that means about what the cells are actually detecting, I'm going to hold off on that question because that's what the first set of data is directly meant to address. Okay.
Yeah, go for it. Thank you.
All right. Okay, so another cell type that also responds, and this is very important in the context of sort of wound response, is the neutrophil, which is the fastest moving of the white blood cells. So this shows some experiments that Nathan Beliveau has done recently, taking immortalized cell line that behaves like human neutrophils called HL60, and then embedding them in three-dimensional matrices made out of collagen. So this is now a much more sort of physiological environment for cells to move on than either the agars that we saw the physarum on or the glass slide that we saw the keratocytes on. And here in this case, the cathode is over to the left, and you can probably get a sense as you watch the movie that there is a general tendency for the cells to move more or less towards the left. This becomes a lot more clear when Nathan actually tracks the cells. So the image on the left of these two graphs is cells moving in 3 dimensions without an electric field. On the right is when the electric field is imposed, and you can see there's now a very strong bias in the migration direction. And because Nathan has set this up in a way that he can just alter the strength of the electric field basically just by turning a dial, you can also see that the strength of this response, as shown in these rose plots at the bottom, increases with increasing electric field. And these kinds of magnitudes of electric fields on the order of a couple of volts per centimeter this is the magnitude of electric field that's actually been measured in both mammals and fish as being the sort of magnitude, field magnitude that happens across wounds. Okay. So let's put a little more detail of what the cells are actually when they detect the presence of neutrophils. So for neutrophils on the left, they're moving in this three-dimensional matrix in a very complicated way. And when the field turns on, which you can see by the little red symbol at the top, what you'll see, as the thing loops back around, if you pay attention to the cell right in the middle, is it seems to withdraw the protrusion that's pulling it in the wrong direction and extend a new protrusion in the direction of the cathode. So that sort of makes sense that, the cell can somehow detect the electric field, put out a new protrusion so that's heading the right direction. What happens with the keratocytes is a little bit different, and I think really interesting. So here you'll see an arrow that indicates when the electric field is on. And what happens here is instead of putting out a new protrusion, the keratocyte actually swings around so that it's able to follow the electric field. And then when the electric field turns off, it sort of forgets what direction it was going, and then it sort of drifts off in different directions. So it seems like in both cases, they're able to detect the field. For neutrophils, it is encouraging them to make a new protrusion. But for keratocytes, it's somehow altering the balance of their overall mechanics so that they're able to make these smooth turns. So this was something that Greg observed, as soon as he started setting up these experiments to look at the keratocytes on the microscope. And he was really intrigued by that question of how the electric field is able to essentially alter this left-right imbalance of the mechanical elements that are contributing to keratocyte movement so that you get these smooth turns. And so he set about then dissecting this mechanism by doing specific tests of multiple different hypotheses that have been proposed in the literature over this past 100 years as to what it is that the cells might be detecting. And this, I partly wanted to tell this story because I think it's a really nice illustration of the overall PBOC philosophy, which is that ideas have consequences. And so when you have a hypothesis about how your phenomenon is working, you should be able to figure out what are the consequences of that hypothesis, and then design experiments so you can test quantitatively whether or not those consequences are in fact true. So for the sort of four most popular kinds of models that people propose for galvanotaxis, Greg drew this beautiful illustration of all of them on the context of the same cell. So by far the most common mechanism that have been proposed is shown up here in part A, which is the idea of asymmetric ion flow in the context of an electric field, and specifically the opening of voltage-gated ion channels. So we know most cells have these really elegant voltage-gated ion channels. They're particularly familiar in the context of things like action potential propagation in neurons or light detection in photoreceptor cells. The idea is that across the membrane of any cell, you also have an ionic imbalance, and changes in voltage in that transmembrane voltage can cause ion channels to open or close. And if you put a cell in a DC electric field, given the magnitudes of the fields that cells respond to, which is a few volts per centimeter, what that means is the difference between what a cell is experiencing at one end versus the other is a difference that is on the order of a few millivolts. Now that might not seem very much, but that is also, certainly the amount that can detect a single photon in a photoreceptor cell. So it's a plausible biological range for changes in imbalances in the opening of voltage-gated ion channels to be influencing galvanotaxis. So if you look at, proposed hypotheses, I would say 90% of proposed hypotheses fall into that category. There were some other things that were floating around out there, though. For example, one that I found very intriguing was part B here, which is electroosmotic fluid flow. And I didn't know this before Greg started working on this project, but this is actually how capillary electrophoresis works. So the idea is if you have a charged surface, like a piece of glass, then all the salt ions in solution will not be totally uniformly distributed. If your charged surface is negatively charged, like glass is, then there'll be a little Debye condensation zone where you have a slight enrichment of positive ions that are right next to that surface. And so what that means is if you then put an electric field across that piece of glass with those positively charged ions that are in its vicinity, the positively charged ions, the cations, will move towards the cathode. And because you've got this sort of spatial segregation of enrichment of ions in different locations with respect to the surface of the glass, there will be a fluid flow along the glass that's heading towards the cathode. Now, if you imagine a cell that's sitting on that glass, this electroosmotic fluid flow phenomenon is over very short distances. It basically means you're going to have rapid fluid flow underneath the cell, heading towards the cathode. And so you can easily imagine that a cell would be able to respond to that sort of fluid flow because they can respond to force, they can respond to shear, they can do, plithotaxis. So it might be that this is just a form of essentially stress sensing that comes about because of this electroosmotic flow. Now, C over here indicates that the electroosmotic flow, of course, applies not only to the surface of the glass, which is charged, but also to the surface of the cell, which is charged. And so similarly, because there are a lot of particularly highly charged sugars along the surface of the cell, you can get the same sort of effect. And then if there are proteins that sort of have extensions that stick out in that fluid flow, that could also act as a mechanical force sensor that maybe can give the cell some sense of direction. And then finally, part D down here is maybe a little less exciting, but it's certainly something that must happen and that the cells have to deal with in some way, which is that different proteins on the surface of the cell have different charge constituencies. And so if you put an electric field across the cell, you might actually get electrophoresis, where proteins or even glycosylated lipids in the cell may move in one direction or the other based on what their net charge is on the surface. So Greg set about trying to compare these hypotheses, these different hypotheses, basically by embracing this idea that ideas have consequences, and thinking through, assuming each one of these hypotheses were true, taking them one at a time, what would be a definitive experiment that he could do that would actually demonstrate whether or not that particular concept actually had any role in galvanotaxis. I'd like to point out these are not mutually exclusive. Certainly multiple ones of them could be contributing. So what he set out to do was essentially try to eliminate them one at a time by designing really definitive experiments to prove that they did not contribute. Okay. So we're going to start off with ion channels. And like I said, 90% of the proposals in literature were in this ion channel domain. It's by far the most obvious possible mechanism. And so the obvious experiment to do is to mess with the ion channels by messing with the ions in the media. And this is one of the particularly fun and awesome things about working with these fish skin cells is you can do things like dilute the media twofold or increase the salt in the media twofold. And it doesn't matter, the cells are totally fine. And if you try to do that to a mammalian cell, they'll just totally freak out and die. But these fish cells, because fish have to be able to move around a variety of different kinds of osmolarity in their environment, you can actually change their salt concentrations without really having any serious consequences on these cells. So Greg did a bunch of experiments in changing the salt concentration, and the sort of standard way that we did the quantitation was to look at the directional bias for a lot of cells.
Hey, Julie, wait, we're having a hard time hearing you, or at least I am.
Yeah, no, so do I.
Okay.
There you go. Now it's better.
Okay, maybe I just wasn't sitting in a good place. Okay, let me know if that comes up again. Anyway, so over here on the left, this is for cells that are not in an electric field. And basically this polar plot that we're looking at takes all of the tracks of individual cells that he saw in the microscope and then just bends them in an angular sense as to what direction they're going. And so if there's no electric field, they go in sort of random directions. But over here on the right, this is a plot with 10 volts per centimeter electric field. And you can see this imparts a very strong directional bias. So he looked at the magnitude of the directional response, which you can measure just as being essentially the cosine of the typical angle. For normal media, for media diluted 4-fold, and for media diluted 10-fold. And what you can see is, as a function of electric field strength, there's much less of a response as the ions get further and further diluted. So, to a first approximation, maybe that's consistent with the idea that ion channels somehow are playing a role. But he noticed something really interesting also, which is that he could take all of these curves, the green one for full media, the red one for media diluted 4-fold, and the blue one for media diluted 10-fold. And they would actually all collapse onto a single curve if instead of plotting the response as a function of electric field strength, he plotted the response as a function of current. Now, we'll see in a minute why this seems to be very important, but one thing it sort of suggests is that the cellular response is not actually dependent on the magnitude of the voltage drop from one side of the cell to the other. The magnitude of the response is related to the current, the actual ionic current that's flowing by. So that is maybe a little hard to interpret in the context of this ion channel model, but it certainly suggests that voltage-gated ion channels are not going to be the answer, because voltage doesn't seem to be the thing that's determining the response. It's current instead. So the conclusion from this was that cells seem to respond to the applied current rather than the applied voltage. But we didn't really quite know exactly what to make of that in the context of this model. So he came up with a bunch more experiments, and I'm not going to go through the details of this because you're welcome to read the supplemental material in his EPIC paper that was published in Current Biology. But there are lots of other experiments you could do besides just changing the ionic strength. So one really obvious one is to just change the nature of the ions in the media. So because these cells are so tough, you can take away all of their sodium and replace it with potassium. Again, that would kill a mammalian cell. It would also kill you. I mean, if you injected potassium into your bloodstream, it would give you a heart attack. But, the keratocytes are fine. You could also use a lot of different drugs that block various different ion channels that would be expected to be involved in regulating the transmembrane potential and would be candidates for being voltage-gated channels operating the system. And you can also do the opposite, which is to apply ionophores that basically allow free passage of ions, including things like potassium or calcium, across cell membranes. And again, he did the same sort of experiment of just taking a bunch of different tracks. In this case, these are now individual tracks rather than pooled by angle. The left is the control, the right is a sort of normal electric field response. And from, I think it was something like a total of 32 different conditions that fell into these categories, he couldn't find a single one that had any effect at all. So, and this included, I mean, all sorts of crazy things you wouldn't believe. So it's not just, that we didn't find the right drug that hit the right channel. It's really that everything that you could possibly imagine that would affect transmembrane voltage and voltage detection has absolutely no effect on this response. So I think the only rational conclusion from this is that the answer is no, ion channels do not contribute to galvanotaxis. So moving on to the electroosmotic flow. So this one now actually started seeming really intriguing. And remember, there's like sort of two separate versions. There's one where you have electroosmotic flow at the substrate, another where you have electroosmotic flow at the cell surface. Either one of these could in principle be detected by the cell. And fortunately, there's a single experiment that's like super definitive that can actually address both of them, which is to simply reverse the direction of the electroosmotic flow. Now, glass is normally negatively charged. And so if you coat glass with polylysine, you convert that negatively charged surface into a positively charged surface. And so in the context of a DC electric field, this is just going to make the flow go in the opposite direction. It's really straightforward. Greg was able to actually measure the flow profile in the chambers that he was using for these experiments by basically putting in little plastic beads that would just, uncharged plastic beads that would just float along with the water. And what he could see is under normal circumstances, when the cells are moving towards the cathode, that it is true that right at the substrate that's shown down here, there is net fluid flow towards the cathode. There's also net fluid flow at the ceiling, because in these chambers, the ceiling was also made of glass. And because of that, and because the chambers were closed, there was net rearward flow in the middle, just so that everything would stay balanced. So in the control profile, the cells move towards the cathode, there's fluid flow towards the cathode. And this is just to show you sort of the relative height of the cell compared to the chamber. After coating the substrate with polylysine, this was the measured flow profile. It goes exactly the opposite direction. So it's a really simple yes or no question. So the cells turn around if you reverse the direction of the electroosmotic flow? And they don't. They don't even think about it. So again, the conclusion clearly is no. So this is pretty good. We're slaying hypotheses left and right. And the only one we have left is this idea that there might be electrophoresis of membrane components that can contribute to galvanotaxis. So there are a couple of different ways that Greg came up with the idea of actually testing this one. Again, the only thing we really think we can do is rule something out, but we can certainly make predictions about, if it were true, what kinds of things would we expect to see. One thing we would expect is that if we alter the pH in the medium, then we would change the net surface charge on components of the membrane. And that might change at least the magnitude of their propensity to electrophoresis in one direction or the other, or even possibly change the direction in which they electrophoresis if we can really get them to, sort of switch charge by changing pH. Again, for mammalian cells, you don't have a very big range over which you can change the pH. But for these fish cells, it turned out Greg was able to change the pH over an astonishingly large range from pH about 5.5 up to above 9 without really changing the speed of the cell. So the speed of the cells at these different pHs is shown here in red. You can see, there's a little bit of variation, but not really very much. On the other hand, the directionality in the electric field is strongly affected by pH. So for anything above pH 6.2, we're close to this complete response where they're always heading towards the cathode. But as soon as we drop down to pH 5.8, now there's essentially no directional response at all. In other words, titration of the pH, which is going to change essentially the protonation of residues on the surface of proteins in the membrane, and also change the protonation of the sugars, either on glycosylated proteins or glycosylated lipids. Once you drop below this critical value, seems to make it so that the cells can't detect the electric field at all. So this is at least consistent with what we would predict from this model. Certainly, clearly, we cannot rule it out. Okay, how about some more experiments? So this is really clever. So Greg came up with this idea that if the way the cell is detecting which way it should go is by electrophoresing membrane components to one side or the other, that there should be some time scale associated with that. And you may remember that movie I showed you before where you take a keratocyte and put an electric field across it. There is a time scale with which the cell is able to respond and change direction. And that time scale can be measured. And so Greg's hypothesis was that if the time scale effectively reflects the fact that these membrane proteins have to move from one side of the cell to the other, then if you do something that slows down that movement, then that should affect the time scale of response. So what he did to slow down the movement was just change the viscosity in the medium by adding methylcellulose, which is a viscosifying agent. The idea being that having a really high viscosity would sort of slow down the movements of the proteins because, of course, they all have sort of elements that are sticking up into the medium as well. And so he was able to measure the time to respond when an electric field turns on, which for a cell just in regular medium happens very quickly here in just a couple of minutes before it gets to a steady state, but is much slower with methylcellulose in the medium, increasing the viscosity. He could also measure the time to forget. So after you shut off the electric field, how long does it take before the direction of the cell becomes random again? And here, again, you can see that it goes fairly quickly in regular medium, but is substantially slowed down in the presence of methylcellulose. And this just shows this effect is very robust with respect to different field strengths, 1 volt per centimeter, 5 volts per centimeter. And the fact that there's the same effect on the time to forget as there is on the time to respond, I think is really intriguing, because this indicates that this viscosity effect is not just something about, the cells are unable to detect or something like that, but rather that once they're polarized in the electric field, their time to forget is also slower because it's slower for things to diffuse away after they've been polarized. So that also is at least consistent with the idea that this might be the mechanism. And then finally, of course, we want to ask if it were just possible to observe electrophoresis of membrane components in these cells under these conditions. And so the experiment he did was to take a fluorescent lectin, concanavalin A, which just binds to sialic acid residues on the surface of cells, and then look at what happens when you put an electric field across. So for this cell, you can see at the beginning, it's heading over to the right. So this is its lamellipodium. The con A is sort of distributed in kind of a speckly way over the surface of the whole cell. But if you keep your eye on this right edge of the cell, as the movie starts playing and the electric field comes on, you can see there's an enrichment of con A molecules all the way over there on the right, which is equivalent to enrichment at the anodal side, that is, as the cell is being told to turn in the opposite direction. So this is something you can quantitate just by looking at the distribution of intensities. So this is, for example, for a cell with no electric field, looking just at the fluorescence intensity from close to the nucleus to the periphery at 2 different times, and then one where there is an electric field, looking at the difference before and after the electric field is turned on. You can see there's a really strong enhancement of the con A on the anode facing side of the electric field after it's turned on. So again, that all seems to be consistent with the idea that this could be the underlying mechanism of gut biotapsis. And certainly, nothing that has come out since this time has suggested that this can't be what's happening. We still don't know, of course, like what the proteins are that are most important in this response. The fact that you can get this reversal response with things like the PI3 kinase inhibitors suggests that it's not just like all the proteins that are playing into this, but that there's some important subset of proteins that's able to couple to those signaling pathways. But we don't yet know the identities of those proteins. But let's get back to the question that somebody asked a little bit earlier, which is, is any of this physiologically relevant? Is any of this relevant in a living animal? And this now is work by my current graduate student, Andrew Kennard, who single-handedly in my group spearheaded our movement from primarily studying migration of isolated cells in tissue culture to now being able to actually study cell migration in living animals. This was entirely his effort from the get-go. So we knew, because of Greg's work, all of these details about what the cells do and don't respond to after they've been isolated. And so Andrew had this very ambitious plan to actually be able to look inside a living animal and see if directional responses for cells migrating in vivo actually would follow the same kinds of cues as what Greg was able to demonstrate in vitro. So this just shows you a diagram of the bilayered epidermis of a zebrafish larva. If you were to take a slice through part of the tail fin here, what you'd see is there's an underlying matrix, and then there's 2 layers of epidermal cells. There's superficial cells that form tight junctions that actually are the sort of waterproof layer that keeps the fresh water out of the animal. And then there are these basal cells that sit right on the extracellular matrix, and at least at this stage in embryonic development, are just a single layer between the matrix and the superficial cells. And this cross-section here is a beautiful thin section electron micrograph that Andrew took, slicing vertically through this. You can see these superficial cells have these really neat ridges on them, and that's actually a story for another time. Here you can see the tight junctions, and then you can also see down here below this other layer of basal cells. And we knew from molecular markers that the cells that we isolate in culture actually are these basal cells. So Andrew, with a lot of help from some wonderful friends at EMBL and at UCLA, Darren Gilmore and Alvaro Sagasti, set about making a series of zebrafish lines where we could express markers specifically in those basal cells, those keratocytes, and then see what they actually do inside of an animal. So looking just at the tail of one of these animals, it's now expressing LifeAct GFP, which binds to actin filaments, expressing it only in the basal cells. What you can see is when you just take a little embryo that's minding its own business, and I'm going to recall this, and you look at the LifeAct in those cells, you can see there's a little bit of dynamics in this epithelium, but as you'd expect for an intact epithelium, there's not really any migration. The cells themselves aren't going anywhere. When we isolate them, we break up the epithelium, we take away all of the neighbors. So the rapid motility that we see with isolated cells is actually thought to be sort of a wound response, is what you would expect for epithelial cells when you take away all of their friends. And so we thought, well, even though under normal circumstances, there's really just not a lot of movement going on in this epithelium, that this might change if they were wounded. So Andrew developed a wounding assay, which is cartooned over here on the left, basically to imitate what would happen if somebody came along and tried to chomp on the tail of the little baby zebrafish. And this now is the same animal, or not the same animal, but the same zebrafish line as you saw in the previous movie. But now after Andrew has come in with essentially a needle and ripped through its tail fin in these 2 locations to cause local tissue damage. And the time is going to play up here in the upper left-hand corner. So you can see here now, in contrast to the previous movie where the cells were very quiescent, as soon as this damage is caused to the tissue, all of the keratocytes in this basal layer now become very polarized. You can see enrichment of fluorescent actin that's heading towards the direction of the wound. And then the cells over here in the epithelium, really within a couple hundred microns of the wound, so really quite a far distance, all polarize and start migrating towards this place of tissue damage, with their goal being, of course, to close up the hole. So it's a little hard to see what's going on with individual cells when you have every single cell labeled. So Andrew also made some animals where they're labeled in a mosaic sense. So only every few cells will express the LifeAct GFP. And so you can see here down in the bottom, this is what happens to an individual cell right after that wound. You can see it polarize and then crawl over towards the right. And then eventually it is able to stop and get back to the point where it's able to regain its normal epithelial organization. So this is now a great system for studying things quantitatively. It's extremely reproducible from one animal to the other. The time it takes is on the order of sort of 15 to 30 minutes, which is a really convenient time scale for studying by video microscopy. And it's clearly involving polarization, migration, and then cessation of migration for these keratocytes, whose in vitro behavior we understand very well. Now, it's sort of interesting to note that although I've shown you lots and lots of images of keratocytes crawling on in vitro, this keratocyte in vivo actually doesn't look at all the same. Its shape is totally different from the shapes of the cells that we're used to after they're isolated. And that also is a story for another day, but it does raise the concern that, you know, a lot of the things that we're able to see and measure and characterize so well in culture aren't necessarily going to apply directly to the animal. Okay, so so how are we going to get to electric fields? Well, the place where this started actually was because in the context of wound healing, there had been this really beautiful paper published in JCB from Philip Nietheimer's lab that showed that you could alter the wound response by altering the tonicity of the medium, specifically by replacing the usual hypotonic or sort of freshwater medium that's used to culture zebrafish embryos with isotonic medium where you had high salt. And remember, I showed you in the context of isolated cells, you can change the salt and it really doesn't change the response to an electric field. So what about the wound healing? Well, if we put the fish in this isotonic medium, this is the kind of typical response that Andrew would see. So over on the right, whoops. Okay, I'm not sure if we can play it again. So let's look at the movie over on the right. There is a little bit of contraction right around the wound margin, but you don't see that massive sort of polarization and migration that, as I described before, that you see standardly in the hypotonic medium. So it really does seem like, in this case, changing the salt condition changes the response quite a lot. And that's illustrated quantitatively over here on the right. The blue line shows movement for an unwounded fish. The black line shows movement for a fish in a normal medium environment. And then this red line shows what happens when you put in an isotonic salt. So that's pretty interesting. That already seems pretty different from what's going on in vitro. And this is exactly what had already been described by the Nietheimer group. Okay, so the model then is that hypotonic shock is an injury signal, that cells are somehow detecting the presence of the wound because of a change in salt, that individual cells aren't sensitive to, but the tissue as a whole is sensitive to. So first, Andrew just wanted to determine whether this was because of sort of local changes in osmolarity around the cells at the wound, or whether it might have something to do with global freshwater physiology, where if you put a freshwater fish in a salty solution, it's not only going to affect, you know, sort of the cells that are right next to the wound that you're going to make, but it's actually also going to affect all the physiology of its gills and its skin and all of its normal ion transport mechanisms. And in fact, if you leave a zebrafish in, you know, salt water for a long time, that zebrafish is not going to survive. So Andrew wanted to design an experiment to tell the difference of whether this was a local cue or a global cue. And I'm just going to blast through this so I can get to the.
Hey, Julie?
Yeah.
Sorry. We should take stock of how you want to proceed. So it's 9:57 and there's 20 questions lined up. So I don't know if you want to keep going and like go until 10:05 or something like that, and then field what questions we can, and then consider the rest, which we can send to you, or if you'd rather be interrupted now, or you know, like which I'd prefer you to decide what you think is the best plan.
Well, I've spent all this time setting up this question of whether or not electric fields are important in vivo, so I do want to show you that result.
Gotcha.
There's a lot of intermediate stuff here about, you know, local versus global and that kind of thing. So I'm going to blast through the next couple of slides so I can get to the meat. So the short version here is it's a local response, not a global response. So that's cool. There's something the cells are detecting directly. The work from the Nietheimer lab suggested that it had to do with cell swelling, which Andrew was able to measure directly in a fairly clever way. But then he started trying to do more experiments along the lines of what Greg had done. So rather than just saying, you know, isotonic versus hypotonic, to change the salts, and not just change the concentrations of the salts, but change the identity of the salts. And once he started doing this, that was when things started getting really interesting. So again, if you just do this in sodium chloride, then the cells don't respond. But if you do it either in hypotonic medium and regular medium, or an isotonic medium where the salt is something other than sodium chloride, you do get a response. It's not as much as you get in the normal sort of freshwater medium, but it's way more than you get in sodium chloride. So this just shows the quantitation for that. This is now, instead of just using sodium chloride, using choline chloride, sodium gluconate, something else I can't see here because it's underneath the box. But you know, doing a bunch of different salts, it doesn't get your full response as what you have in freshwater, but there's a lot more response than what you get in sodium chloride. And this has nothing at all to do with that osmotic shock with the cell swelling, because really none of these things transduce a lot of cell swelling, although a lot of them, choline chloride, sodium gluconate, et cetera, do give you a migratory response. So there's something special about sodium chloride that's different from the rest of the salts. And this gets back to this issue that I described in a sort of hand-wavy way about the transepithelial potential. So there's going to be channels and pumps on the apical versus basolateral sides. There's also leaks. And, ooh, that didn't work. And anyway, because of the way that this is set up in the zebrafish epithelium, you expect that there's going to be a transepithelial current that gives you a transepithelial potential. And this has been characterized to some extent in zebrafish. This is a vastly oversimplified diagram. There's actually lots of other channels that are involved. But both sodium and chloride establish this transepithelial potential, whereas some of these other salts like, gluconate, choline, these are not things that are really major ionic elements in the fish's normal environment. So the idea is, as I mentioned, that if you have a wound that sort of short circuits this transepithelial potential, and you go from having a significant measurable epithelial potential on one side to having a short circuit where the voltage is 0, and that automatically gives you an electric field where the cathode, that is, the negative pole of the electric field, shows you where the wound is in the direction of the wound. So Andrew then did some really pretty hardcore experiments where he took one of these fish and just stuck in some electrodes and then was able to turn on the electric field. And this is now an unwounded fish, and he stuck in the electrodes very gently and then sort of waited until everything had settled down from a tiny injury. And then was able to actually switch the electric field back and forth. Let me just play this movie again. So here, this is before the electric field. Now the field is heading towards the left. You can see the cells moving towards the left. And now they're heading towards the right, and you can see them moving towards the right. And he could track this movement of the cells in what's now an intact epithelium where there's no wound, heading towards the cathode in each of these cases where the electrode is impaled, and he could just switch the direction of the field by flipping a switch. And this just shows you some quantitation, again, that doing this experiment several times, the cells always move towards the cathode, and when you switch it, they change back and move the other direction. Okay, so far we know that cells respond to a wound in a way that is consistent with them having to detect something consequent to the transepithelial potential. That is, there's something special about sodium chloride that's not reproduced by other salts that are able to give you the same osmolarity. And we know that cells in the tissue can, in fact, respond to an electric field if it's imposed externally with these electrodes. And so then the big question is, well, is the electric field actually signaling in the context of the wound? And that, again, is something that's very, very hard to prove. But again, if it were true, then the prediction would be that if you take a wounded fish and set up an electric field that's heading in the wrong direction, you should be able to override the normal directional response of the cells. So that was this incredibly technically difficult experiment that Andrew was able to get to work actually just very recently, just a couple of months ago, where he was able to do the wounding and the electric field application simultaneously. And you can probably see it best over here in this graph, where the black lines here show the response, the motility response of the cells within that couple hundred microns of the wound. In black, under the normal case, the unstimulated case of just wounding. And then in blue, when he imposes an electric field that goes in the opposite direction. So you can see is not only do the cells, after sort of an initial response, not only do they slow down, but they actually end up going in the wrong direction if that electric field is superimposed the wrong way. So this is now our current model. Previously it had been suggested that hypotonic shock induces cells to migrate, and there certainly is a component of that. But what Andrew has discovered is that there's a separate effect that has to do with this transepithelial potential that's generated because of the physiological transport of sodium and chloride across the fish skin that leads itself to actin polarization in a way that's completely independent of this osmotic effect. And that actin polarization gives you not just migration, but actually migration in the correct direction. So there's lots more interesting things to do here. Obviously, we're super interested in understanding not only how this electrical effect gives you directionality and wound healing, but also how it interacts with the mechanics. So these cells are in epithelium, so they're also pulling on their neighbors. And so we might well have other kinds of effects, mechanical effects, that enable these cells to retain their shape and remodel their shape as a function of the response to this wound healing. And again, I'll just go back and play this movie again. There's a lot of really fascinating things going on here in terms of the cells moving, and then how do they know when to stop? And then after they've stopped, you see this thing where they all pull apart from their neighbors. So how did they know how to do that? And then how did they go back to being a normal intact epithelium? So I think really in order to dissect this, we're going to have to deal with all of these mechanical issues as well as the electrical issues and signaling issues that I've described today. All right, so that's where I'll start. So the first set of experiments that I described on breaking down those 4 hypotheses for electric field detection were done by Greg Allen when he was a graduate student, and that was in collaboration with our long-term friend and collaborator, Alex McGillner. The initial work I showed you on migration of neutrophils in 3D matrices was all done by Nathan Beliveau, and then the in vivo work on the contribution of the electric field to wound healing in real live zebrafish embryos was all done by Andrew Kennard. So it is 10:05, so I will stop there and I'd be very happy to now start taking more questions.
Thank you, Julie. That's super fun. Great to hear that story, which I hadn't really heard. So there's quite a few questions. Many of them came from the first part of the talk. So I think Ernan and I are going to try to summarize them and ask them broadly. I don't know. You want to give it a try, Ernan?
Yeah. So there are a few, and so one has to do. One is the basal cells don't have tight junctions. So how do they modulate transepithelial potential?
So the basal cells don't have, at least they don't have very strong tight junctions, but the superficial cells that are above them, the peridermal cells, those do have tight junctions. And something I left out, actually, Andrew gave me a diagram of this, but it was just so terrifying I couldn't include it. There's also this whole set of cells that are called ionocytes, of which there are something like 4 or 5 different classes that have been identified in zebrafish that actually span both layers. And so the transepithelial potential is set up by a combination of the action of the superficial cells, which do have tight junctions, and then also these ionocytes that go the whole way. And the 4 different classes of ionocytes are distinguished by the fact that they have different suites of ion channels and pumps on the apical versus basolateral surfaces. So Andrew started looking into this because we thought, oh, we can just, we can knock out the appropriate ion channel and then change the transepithelial potential. And it turns out we can't. It's insanely complicated. But I left out a lot of those details of fish physiology. Of course, the transepithelial potential is maintained not only across the skin itself, but also through ion exchange in the gills of the fish. So there's just a lot that the fish are doing in order to maintain that potential. It's not the basal cells that are themselves responsible for setting up the potential. We think they're only in the business of detecting it when it's been disturbed, not actually setting it up directly.
So
something
go
ahead.
I can summarize. There's probably 3 or 4 questions that have to do with the viscosity tuning knob, which you actually did talk about. And I don't know if you want to elaborate any further. For example, Roya was curious about whether viscosity would change any other aspects of cell motility. For example, the turning to be faster or something like that. And
turning.
a couple other people were asking just in general. Kat had also something about pH. So anyway, that's kind of, all those are classes of questions having to do with the knobs that you might access.
So for viscosity and pH, we didn't actually thoroughly explore what their effects would be on turning. And I think that's actually an interesting and relevant question. The simplest thing to measure is just the speed. And over the range of perturbations that we did, we didn't have any effect at all on cell speed. And for viscosity, certainly that kind of makes sense. I mean, the cell, like I said, we're super low Reynolds number. So it's always in some sort of state of force balance all the time and always generating active force in order to have any velocity. And in that case, really the main constraints on the cell's ability to move are the fact that it's making strong contacts with its substrate, through integrins onto the glass surface. And so changing the medium viscosity, is just a tiny, tiny perturbation as far as the actual sort of resistance that the cell is encountering. Because its Reynolds number is already something ridiculous, like 10 to the minus 4th, and so you're making it into something like 2 times 10 to the minus 4th. It just doesn't matter. And likewise for the pH, certainly you can imagine that there would be effects of altering the protonation state of every molecule on the surface of the cell. You'd think might bother it, in some way that would affect its motility. But again, at least as far as the speed is concerned, it really didn't seem to have an effect over that range. I showed the range of sort of 5.5 to about 9 because that's the range where it doesn't have an effect. If you go way higher than 9 or way lower than 5.5, then you start seeing the cells lose polarity and eventually die. But at least over that range, it doesn't seem to bother them. So I do want to emphasize though that sort of speaks to the importance of choosing the right system for the question that you want to study. because if we were to try to study this in human neutrophils, all of those experiments would have been impossible. You can't, culture a happily motile neutrophil at pH 5.5 or pH 9. The fact that the fish are able to deal with that, I think, reflects, sort of the selection that they've been under through evolution to be able to deal with, rapid changes in their environment in a way that mammals, we don't really know how to do that, because we carry our saltwater ocean inside of us. And so we're just not that good at dealing with things like changes in pH or changes in ionic strength. So at least for the keratocytes, that's definitely within the range that they have no problem dealing with normal physiological responses. But those are certainly important considerations.
Thanks, that was helpful.
So one that I thought maybe you touched on this, but I thought it was worth bringing it up. So what is the behavior of the outer peridermal cells during the wound closure? Do the basal cells get polarized first and transmit the cues to the outer cells, or vice versa?
These are great questions. So Andrew and then several other members of the sort of zebrafish crew in the lab are now trying to look not just at the basal cells, but also at both cell layers simultaneously and the coupling between the two. As far as we can tell, just from sort of preliminary imaging, the peridermal cells seem to be pretty passive. So it seems like they maintain their tight junctions with their neighbors. They do get stretched, but we don't really see the peridermal cells changing their relative locations with respect to the basal cells. So at least as far as we can just see by eye, by trying to track the two layers, it really looks like the basal cells are calling the shots in terms of this active migration towards the wound. And then they're just dragging the peridermal cells along with them and consequently stretching the peridermal cells. They're setting up now. So this is Andrew together with, actually they're all right here. So this is Chris, this is Ellie, and this is Mugda. So those four there are the zebrafish team. And they're also trying now to set up lines where we can actually start looking at junctions, not just between the cells within each layer, but also the junctions between the two layers. So we can get more of a sense of how all those rearrangements are happening as the skin is made.
One quick question from Yaya, one of our alum, is the other proteins are not present in the keratocytes in the context of a tissue, but are expressed when the cells are on their own. I mean, this is about the difference in behavior when they're by themselves or in the tissue. Yeah.
So when we isolate the cells, we do these experiments typically within a couple hours after isolation. So there's probably not a lot of time for the transcriptional profile to change drastically. The two other bits of information about that. One is Sunny Lowe, who was the first member of my group to actually culture keratocytes from zebrafish. She did bulk RNA transcription studies on cells, sort of at various times, isolated at various times post-fertilization from embryos. So she has a whole catalog of what the genes are expressed in isolated cells. And then in the meantime, Alvaro Sagasti's lab at UCLA had done quite a comprehensive RNA sequencing based study on gene expression in the different layers of skin and also a number of other tissues throughout the fish. And they look pretty similar as far as we can tell. It doesn't mean that there might not be a couple of critical things that change, but there's certainly not a big global change that happens over that just first few hours. Now, if you try to keep the cells in culture for many days, then over long periods of time, over days and weeks, then you do start seeing a lot of changes in gene expression that accompany changes in cell behavior. So they go from having this really epithelial characteristics and the rapid migration of isolated cells to switch over to a more sort of fibroblast-like phenotype and fibroblast-like behavior, and that's accompanied by long-term changes in expression. So we can't rule out specific things, but just the time scale, I think, suggests that that's probably not the major reason for the difference in behavior.
So there's still quite a few questions, but I'm going to be democratic and just choose the next category where there were the most questions on the same thing, which is, and then this we'll have to wrap up after this. So there were several people that were curious about magnetic fields.
Oh, yeah. So magnetic fields and electrical fields, of course, are just two sides of the same coin. And some of the snake oil devices that people sell for wound healing allegedly produce magnetic fields. Although, of course, if you're producing a magnetic field, you're also producing an electrical field. We have not made any attempt to explicitly separate magnetic fields and electric fields in these experiments. I'm describing them as electric fields because what we're doing is, we're putting electrodes in, with an auger bridge on either side of porous medium that's filled with a salty solution. So it's very obvious how to think about and how to measure the electric field in that context.
Yeah, but if I understood you correctly, it's DC. So, in that case, you probably are not going to generate any magnetic fields. And I have a feeling, that people are thinking, just straight up static magnetic field, for example.
Yeah. So there are certainly plenty of biological systems that are designed to respond to straight up static magnetic fields. The homing pigeons are a famous example. There's also magnetotactic bacteria that have little, crystals of magnetite that they line up that are able to orient themselves for flagellar-based migration in static magnetic fields. We don't have any evidence that these migratory cells are able to do that. I'm not aware that anybody's actually tested that. If anybody knows of anybody who's tested that, I would love to hear about it. It's not something that we've tried to do.
Yeah. Okay. Yeah, I think it's quite interesting. Yeah. What a great topic in general. Okay, so I think we'll wrap up. So thanks again, Julie. That was really super fun and interesting as usual. And let me just alert everyone to next week's schedule. So on Monday, we have Janne Kondev, who's going to talk about how to build a scale invariant cell. And I don't know, I'm biased, but I think he gives great talks. And then on Tuesday, we have Dan Needleman, which should be fun. And then Wednesday, Lena Koslover from UCSD is going to talk to us, which I'm looking forward to quite a bit. And then on Thursday, Armita Nur Mohammed from UW, who works on very interesting population genetics things. And then on Friday is Thomas Lequi, which also promises to be great. So we have a really nice lineup for the coming week. So I hope the rest of you have a great weekend, and thanks for tuning in, and look forward to seeing everyone again on Monday morning. All right. Anything you want to say, Erna?
No. Thanks, everybody. Have a good weekend. Stay safe. The usual.
All right.
Thanks again, Julie. Great talk.